Hello, welcome to Biology with Mr. Z. Today we are talking about the central dogma of molecular biology. This is a really important concept that I would say ranks right up there with something like evolution in terms of its importance to biology. And today we're going to look at an overview of this concept, and in subsequent lectures we're going to get into some of the details of the different processes involved with the central dogma. Our title today of our notes then can be the central dogma of biology, and the learning intention is going to be specifically today, how does a gene become a protein? You can pause here if needed to have time to write that down. And this is really what we're going to be focusing on for the rest of today and the next week or so, is talking about the relationship between DNA and traits and how it is that our DNA can lead into our traits. So yesterday we looked at some different traits. We looked at behavior traits, physical traits, and some medical traits that we might have. And as we go on from here, we should be able to start identifying how it is that our genes can lead to those traits. All right, if you need more time again to write the learning intention down, go ahead, pause it now. I'm going to keep talking. Okay, so what is dogma? When we say the central dogma of biology, dogma is basically a set of principles that are laid down by some sort of authority as incontrovertibly true, which means without doubt this is true. It happens like this with religion, politics has a dogma, and so on. So biology also has a central dogma, and in this case, the central dogma is all about DNA and how DNA gets turned into proteins. In your notes, then, I just do want you to write down the definition for dogma. If you have the note sheet, it's going to be fill in the blank. Go ahead and get that down, because when we talk about the central dogma of biology, I want you to know that this is a set of principles that we're going to be referring to over time throughout the rest of this course. Okay. This is something that if you were in class, we would talk about. Why do we write things down? What is the purpose for writing or recording things? And so if you are watching online or at home today, what you can do is maybe on the side of your notebook somewhere, write down what is the importance of people recording things throughout time. Why have we recorded things? That's what I want you to reflect on. And again, you can pause and do that now. Take a minute or so and think about what is the purpose of writing things down. Okay, if you have done that, I'm going to keep talking. The reason we write things down and things have been recorded in the past is to remember them. We, back in the day before the printing press, people would go through things like the Quran, the Bible, and the Torah, all these important books, and they would go line by line, and they'd have to write them down perfectly to make a new copy of it. And it was so that we could remember those things. It was something that people felt was very important. We didn't want to forget it, so we had to keep writing it down to remember it. And before the printing press, we had to do that, make copies by hand. The printing press came about, and then we got to do it with machines, and now we can make copies with the Control-C, Control-V. Boom, you have a copy of something. But the reason that we do record things is to remember them so we can access them in the future. And it turns out that DNA is just such a thing. It's our genetic code written down. That means that we can make copies of it so we have it in the future. We can reference it whenever we need, and we need to make sure that it's being copied accurately. Okay, so when we think about how books have been passed down through generations, same thing goes with DNA. We learned that the nucleotides in our DNA are like a language, and it has been copied and preserved through time. And we're going to look at why it is, or the parts of it that are so important today. Here is the central dogma of molecular biology. This, in a nutshell, is, I would argue, the most important process in all of biology. We have DNA at the top here that gets turned into proteins. Yesterday we learned that a particular part of the DNA that codes for a protein is called a gene. So the gene gets copied into a messenger called RNA, and the RNA gets turned into a protein, and then the protein goes out and does all sorts of different work for our cell. It does important things like help us grow, it helps us move. Proteins are responsible for pretty much any significant event in our cell. They help all sorts of different chemical reactions occur. They help us digest food. They help us move around, run, breathe. Okay, proteins are essential for life. And if we did not have a way to make new proteins, well, then life would not be possible. Central dogma is how we get from the directions for making a protein to the protein itself. That's the main message here. And this diagram should be in your guided note sheet, so you don't have to write anything down. Next up, a gene. I'm going to say today, I'm going to make this analogy that a gene is like a recipe. Okay, a recipe for chicken, for example, it tells you what ingredients you need. It can tell you how to make it, but by itself, you can't eat the recipe. If I just take this card here that has my recipe written on it, by itself, it is not a very tasty thing. I have to turn it into the tasty thing over here. And so that is what the central dogma is about. How do we get from the directions to the product? Okay. DNA by itself can't do very much for it. It's just a set of instructions. Set of instructions by itself, not that great. What we need are some other molecules or ingredients to get brought into this role. And we also need other players to actually help make the proteins, help make the recipe. DNA by itself cannot make a recipe. It is the recipe. Okay? That's my analogy that we're going to be working with. So this is how I go about cooking. All right? I've got a box of recipes that have all of my grandmother's sacred recipes. They're written down in her hand, and fantastic cook. Every one of them is delicious. Okay, I got them in a box in my kitchen, my mom's kitchen actually. And so when I want to make a recipe, I have to go in and find the recipe card that I want to make. But since these are my grandmother's sacred recipes, I can't ruin that recipe card. If I mess up that recipe card in any way because I spill tomato sauce on it or I accidentally light it on fire because it got too close to the burner, I would ruin that recipe forever and I'd lose my grandmother's recipe. Okay? I can't have that happen. So what I do instead is I call up my mom when I want a recipe and I say, hey, can you look through grandma's recipes and make me a copy of that recipe? So she'll copy it down on a separate card, and that way, and then she'll give it to me, and that way I can take it into the kitchen, I can get tomato sauce on it. If it starts on fire, it's not a big deal because we still have the master copy in the recipe box. All right, so I take the recipe from her and then I make it. I just go through the directions, I gather the ingredients, I put everything together, and I make the recipe. This up here, this is not me, unfortunately. In the end, I end up with a delicious roast chicken meal that is just like my grandmother's. And since I have the recipe now, I have the recipe card or a copy of it, I can make it as many times as I want. All right, and believe it or not, the way that I go about cooking is almost identical to how a protein gets made in the cell. So let's take a look. Similar to the recipe box in our cells, if this whole big blue thing here we're thinking is a cell, the darker blue section is going to be the nucleus where all the DNA is kept. And it's kept in the nucleus to stay safe. Just like the recipe box keeps grandma's recipes safe, so too does the nucleus keep our DNA safe. If a gene gets turned on in our cell, in the nucleus, it means that RNA is copied from it. So RNA is basically just a copy of DNA, and in particular, it's usually a specific gene that gets copied. So here we had our DNA strand, it got copied into an RNA strand, and that RNA strand is now free to move out of the nucleus into the kitchen of the cell, where it can encounter all of the various things that might be dangerous to DNA, but RNA doesn't matter because it's just a copy. We still have the master copy back in our nucleus. All right, the RNA leaves the cell. It is then going to go out into the cytoplasm where it encounters something called a ribosome. And a ribosome is like the cook, in my analogy, that helps create proteins. So it reads through this recipe card, which is our RNA strand, and it's going to make proteins. In this case, I have it making some hemoglobin, which is a protein that helps carry oxygen in our blood. We then have to put multiple hemoglobin subunits together to get hemoglobin. And this is like our final dish in the recipe box. So again, we go from DNA starting in the nucleus. It gets copied into an RNA strand. A gene will get copied into RNA, which is a messenger that can leave the nucleus, go out into the cytoplasm, where it interacts with the ribosome, which is the cook that creates the proteins by reading through the recipe, creates that protein, and then the protein can go out and do its job. Okay, so again, to just compare, the recipe box is like the nucleus, it's DNA in the nucleus. The recipe cards are individual genes, which are individual instructions for proteins. When I had my mom copy down the master copy into a copy I could use in the kitchen, she was turning it into RNA. All right, this is similar to DNA going to RNA. She's transcribing that card. Then I went and I made the protein by looking at the recipe. This is like a ribosome making proteins. And then last but not least, we have the final dish being made, which is going to be our final form of the protein. And again, we can use this recipe card as much as we want, just like in kitchen. We can use an RNA template as many times as we want. So a single RNA could make tons and tons of copies of different proteins. Well, the same protein, but many copies of it. That's the analogy I have for you today. Here is an overview of all of the different processes that are going to happen during this, or in the central, that are part of the central dogma. We have DNA being stored in the nucleus, and it gets transcribed. That's the term that we have for DNA getting turned into RNA. It's called transcription. There's two main processes to the central dogma that we're going to talk about today: transcription, DNA to RNA, and translation, RNA to protein. So transcription happens in the nucleus. It is done by a protein called RNA transcriptase, which basically just goes through and reads off this DNA and creates a copy of it, but it's an RNA copy that is free to leave the nucleus into the cytoplasm. And the purpose of doing this is so that the RNA, it keeps the DNA safe. That's the purpose. This way the DNA can stay safe in the nucleus, and the RNA goes out into the cytoplasm where it might face different toxins or enzymes that want to chew it up, but we have and are able to preserve our DNA strand. It then gets translated through this translation process where RNA gets turned into protein. Here we're going to have a helper called ribosome. The ribosome is a very important protein that's going to read through this transcript, this RNA transcript. It's going to read through it piece by piece and pull in different components to make a protein. And the purpose here is that proteins are essential for life. Without proteins, we just simply could not exist. So it's super important that we're able to translate proteins. And in fact, many antibiotics that fight off bacteria could interfere with any of the steps within this process. Maybe it stops RNA from getting made, or maybe an antibiotic might stop proteins from getting made in bacteria, and then the bacteria die and we get healthy. So when you're taking antibiotics, it's generally messing up one of these processes within the central dogma here. Okay, you have a chart in your guided notes, and I want you to get this information into the chart. Transcription occurs when DNA is turned into RNA, and it's helped along by RNA transcriptase. The purpose of this is that RNA can then leave the nucleus and enter the cytoplasm where it's more dangerous for a molecule to be in existence. So it keeps the DNA safe. It doesn't have to go out into the cell. Translation is when RNA gets turned into protein, and its helper is the ribosome. Last but not least, we have proteins, which are more complex and can do a number of jobs for the cell. The protein is essential for life. It does so many things for our cells that we could not exist without it. All right, this video is going to be posted in the slides, so I want you to go through and watch this because it shows in real time what it looks like when these processes are occurring. And then last but not least, I want you to do these things. First, on the left side of your page, that empty page, I want you to draw a cell and show me each of the stages of the central dogma. I want you to show me transcription, DNA getting turned into RNA, and I want you to show me RNA getting turned into protein through translation. You should include DNA, RNA, protein, transcription, translation, RNA, transcriptase, and ribosome. One thing to note, well, I'll get back to that, never mind. Then I'm going to have you write a story. This description page, this is going to be posted online underneath the video today. So if you have any questions about it, you can reference that. For right now, though, after watching all this, we're going to go back to the beginning, and I hope you can answer this question. How does a gene in our DNA get turned into a protein? There's two steps to it. Transcription, the gene gets turned into RNA, and translation, the RNA gets turned into a protein. That's the answer to this question, and I want you to know it at that level right now. As we go forward, we're going to learn more about transcription and translation and the exact things that are happening in those processes. But for right now, that's all we're concerned with. All right? So thanks for listening today. Have a great time. If you have any questions, let me know, and I will post all the further directions below in the lesson.